Hello students, today we will discuss about Agrobacterium tumefaciens and the vector made from TI plasmid of the Agrobacterium, which can be used to transfer the desired gene into the higher plants. So let's first discuss what is Agrobacterium tumefaciens and why it is known as nature's genetic engineer. So Agrobacterium tumefaciens is a soil-borne bacteria and it contains a plasmid which is known as TI plasmid, and this TI plasmid is tumor-inducing plasmid and it can be used to transfer the gene from bacterium to plant cell. So why it is known as nature's genetic engineer? Because Agrobacterium tumefaciens, which is a soil-borne gram-negative bacteria, it invades dicot plants through the wounds, and through wounds it infects the plant, and there it causes crown gall disease, which is a tumor-like growth. And in this tumor-like growth, it manipulates the plant cells to synthesize opine, which it uses for its own energy. So this is how the Agrobacterium infected plant cell cause disease, cause tumor in the plant cell, and these tumorous cells are transformed, and these transformed cells synthesize opines from which bacterium get its energy, and this is how the Agrobacterium tumefaciens manipulate the plant cells for its own benefit. So that is why it is known as nature's genetic engineer. So before starting, let's discuss why we are discussing it here now, because we are discussing the cloning vectors for higher plants, and one of the strategy, as we know, there is no plasmid present in the higher plants, so we have to use some other type of plasmid which can be used as a vector to transfer gene in the higher plants, and one of them is TI plasmid of the Agrobacterium. The other strategy which we will discuss further will be direct gene transfer and vector based on plant viruses. So here we are discussing about cloning vectors from prepared from TI plasmid of Agrobacterium tumefaciens. So first let's discuss how the disease occurs and how it looks like. You can see these slides. As it is soil-borne bacterium, so it can easily infect the plant near the soil, like in the lower stem. It also causes galls in the branches also, but the more common is near the in the lower stem near the soil. So this type of galls can be prepared, and this is due to the type of gene present in the TI plasmid. We will later discuss what are the gene like auxin and cytokine producing genes are present, which are responsible for causing the tumor. So what happens actually was it is a wound in the if there is a wound in the plant and this wounded site releases some kind of signal, like some chemicals which act as a signals for Agrobacterium tumefaciens, and as Agrobacterium tumefaciens in the soil receive the signal, it invades the plant and cause crown gall, which look like this. So here the bacterium it enters the plant through fresh wound and attaches itself to the wall of the intact cell. There it genetically transformed the plant cell, and this transformed plant cell now synthesize some special metabolites which are known as opines, and these opines are octopine, nopalene, agropine, succinopine, and the Agrobacterium catabolize these opines to obtain the energy. So how a disease occurs is which is responsible what is responsible for the occurrence of the disease is TI plasmid, which is present inside the bacterial cell. Means this bacterium, Agrobacterium tumefaciens, contain a plasmid which is known as TI plasmid. TI stands for tumor inducing. So tumor inducing plasmid is responsible for causing the crown gall disease. So let's discuss about the structure of TI plasmid first. So this is the structure of TI plasmid, and this is a general structure made by me, so that I can explain you the in general category how, because there are specific TI plasmid in which is specify or synthesize the specific opines, and according to that only they are classified and named according to the type of opine they synthesize. Here I am just discussing a general structure in which I have taken this opine synthesis gene present here. So let's discuss according to it. In TI plasmid there is ori, and the one important region is tDNA, that is transfer DNA, and it plays an important role in the infection process, because this is the area which is transferred to the plant cell. So let's see the size of the plasmid. Agrobacterium tumefaciens contains TI plasmid which has a size of nearly 20 kb and 200 kb. So 200 kb, in 200 kb of size there are two essential regions. One is VIR region and other is tDNA. This tDNA region is consists of 15 kb to 30 kb of size DNA, and this tDNA is flanked by two border repeats of 24 to 25 base pairs. This is left border repeat and right border repeat. And inside this, three genes are present. One is for auxin synthesis, other is for cytokinin synthesis, and the third one is for opine synthesis. And this is specific for each TI plasmid. So this auxin and cytokinin genes are responsible for the proliferation of cells and causes and they are responsible for the formation of tumor inside the plant cell in the wounded site. And this opine synthesis gene is responsible for the synthesis of a specific opine, which can be utilized by the bacterium to obtain the energy. And this TI plasmid, which is present in the bacterium, also contain an opine catabolism gene, which is required for the catabolism of opine synthesized by the plant cell or manipulated plant cell. So what is important here is the tDNA, and the tDNA contains the border repeats of 24 and 25 base pairs, and they play a very important role in the transfer and integration of the tDNA. So this portion is transferred to the plant cell. This is important. And how this is important? Because the VIR region of the TI plasmid, which is activated by the signal comes from the wounded site, only recognizes this border repeats. So only recognizes this border repeats, and the gene present inside the tDNA have none of the role played in transfer and integration of the tDNA. So this border repeats is important. So next, the region which is important is VIR region, and this VIR region contains many genes, and up to now 9 VIR gene operons have been identified, like VIR A, G, B1, C1, D1, D2, D4, E1 and E2. So other regions I have already discussed, that opine catabolic region and the ori region. So tDNA is excised from the TI plasmid, then transferred to the nucleus of the plant cell, and thereafter this tDNA is integrated into the DNA of the plant cell, and this integration is stable, and the tDNA can be passed on to the daughter cell as an integral part of the plant cell. Means the tumorous cells which are formed due to the presence of tDNA all may contain the copy of a tDNA. So because the integration was stable, and the daughter cell which are proliferating contain this copy. So this is how we can see in the figure. This is bacterium, and this bacterium when physically get attached with the plant cell at the wounded site or near the wounded site. So this bacterium consists of the bacterial DNA and the TI plasmid, and this TI plasmid contains tDNA as well as the VIR region and other genes also. So this how when the plant cell is exposed and the bacteria get attached with the plant cells, this tDNA get transferred and get integrated with the plant genome, and this is how after the integration crown gall or tumor is caused. So now let's see the signal transduction, how this tDNA is transferred from the bacterium cell to the plant cell. So So this is the wounded site, and from the wounded site some phenolic compounds and so sugars are released, which act as a signal and which activates the VIR region. Here it activates first VIR-A protein is formed. This in turns phosphorylate VIR-G and VIR-G protein is formed. Now this VIR-A and VIR-G together activates the other VIR region and results into the formation of virulence protein like D1, D2, E2, B, etc. So how this work? The VIR D1 and D2 products, or we can say the protein D1, D2 recognizes the border repeats and they form the single-stranded tDNA copy. They are responsible for the production of the single-stranded. They only recognize the D1 and border repeats, and then the single-stranded copy of the tDNA is formed and which is capped by the D2, and together with this the B1 which is formed here is responsible for making the transport apparatus between the agrobacterium and the plant cell. One thing more, the agrobacterium attaches with the plant cell with the cellulose fibers, and these are formed due to the production of the proteins or signals obtained from the genes of present in the chromosomal DNA of Agrobacterium, that are CHV genes, which are responsible for the production of cellulose fibers which are needed to get attached with the plant cell. So here the complex form is single-stranded tDNA along with D2 and G. They move towards the plant cell through the transport apparatus made by the VIR-B. And here as it enters into the plant cell, the products of VIR-E2, E2 proteins get attaches with the single-stranded tDNA, and it covers it to protect it from the action of nuclease in the plant cells. And thereafter this VIR-E2, D2 and the single-stranded tDNA complex is transferred from the nuclear pore complex to the nucleus of the plant, and here tDNA gets integrated with the plant genome. And as the tDNA expresses its gene here, the auxin, the cytokinin production is started, and thus opine synthesis also is started. Auxin and cytokinin gets the stimulation of cell division, and thus the tumor is formed. So this is how the signal transaction takes place, and the agrobacterium get attaches with the plant. Single-stranded tDNA is transferred from the bacterium cell to the plant cell and get integrated with the plant cell and result into the crown gall disease. So up to now I have discussed the process which is followed by the Agrobacterium tumefaciens in nature. In the next class, we will discuss how this property can be exploited to produce the vector which can be used to manipulate plant cells for our benefits or for the production of genetically modified crops. So, we will further see in the next part.